Hi everyone. In this video, we are going to discuss about HIV or AIDS virus, that is human immunodeficiency virus or AIDS virus, and majorly concentrating on discussing about the structure of HIV virus and the multiplication or replication of HIV virus. Whereas we have going to discuss about this AIDS, that means about the disease or the infection caused by this HIV in a separate video. And here majorly we are going to discuss about the structure and multiplication or replication of HIV. So let's see the introduction first, then we'll move to that structure. The HIV, that is human immunodeficiency virus, is a retrovirus and causes a disease or infection called as AIDS. The full form of AIDS is acquired immunodeficiency syndrome in humans. So syndrome is a term that indicates it's a group of diseases. It's not a single disease. And HIV, a lentivirus, which is lenti means slow, belongs to the family Retroviridae. So according to the serotypes, the HIV is going to be of two types, that is HIV-1 and HIV-2. The HIV-2 has reduced virulence as compared with the HIV-1 and causes a milder AIDS-like disease. But coming to the HIV-1, the HIV-1 is supposed to be the American strain as it was isolated in the US. And nearly more than 99% global AIDS is because of HIV-1. Okay, then moving to the structure of HIV. So HIV is a spherical enveloped RNA virus. So HIV is a spherical round shape and it's an envelope. That means it possesses an envelope and the genetic material that it possesses is RNA. So obviously it belongs to the RNA virus. And the virus has a spherical envelope which is about measures of 90 to 120 nanometers in diameter. In some books it is going to be of 100 nanometers also. So that's why on an average 90 to 120 meters in diameter. And now this viral structure is going to have two parts. How many parts? Two parts. The core part, which is cone shaped, encloses this one. The core part, which is going to encloses the riboproteins, that is ribonucleoproteins, RNA containing nucleoproteins. And coming to the outer shell capsid, which is icosahedral in shape. Okay, so these two are going to be major parts of it. One is the inner core protein, that is nucleocapsid, and the outer is going to be of icosahedral in shape. And the genome of the virus is diploid and is made up of two identical single-stranded chains of RNA. And this RNA is associated with an enzyme called as reverse transcriptase. As RNA is associated with the reverse transcriptase, this is the main characteristics of retrovirus. That means the presence of reverse transcriptase is one of the main characteristics of retroviruses. So that's why we have classified this, not we, it was classified under the family called Retroviridae. Then moving, the virus also contains lipoprotein envelope, particularly when it comes out of the host. That means this layer was made up of from the membrane of the host cell. The lipids are derived from the host cell membranes, while the glycoproteins, so these are called as glycoproteins, are produced by the virus itself. That means this envelope was prepared from us, whereas these glycoproteins are going to be produced by the virus. The viral glycoproteins are in the form of projecting knob-like spikes on the surface, and these spikes are going to be called as GP120. Okay, and below these spikes you are going to see some sort of transmembrane pedicel like one, and these are going to be called as glycoprotein 41. So these purple color, all the things are going to be of glycoprotein 120 called as docking glycoproteins also. And the pedicel like that is present to this one, these are all transmembrane proteins which are going to be of glycoprotein 41. So internal to this outer icosahedral shape of the capsid, there is a P818. Okay, inside that, this is going to be of cone-like core. This is nucleocapsid, which is going to be of P24. Okay, and the viral RNA is going to be enveloped by P7 and P9. So these are all the proteins which are enclosing this RNA genetic material. And there is a reverse transcriptase enzyme. So these are going to be of proteins P7 and P9. Then coming to the reverse transcriptase enzyme is also found here. Okay, so this is the overall structure of the virus. So it is going to be of, once again, have a glance of it. HIV is going to be of a spherical in shape, envelope, and is a RNA virus. And as we know, it is going to have some sort of spicule-like structures, which are nothing but the glycoproteins which are attached to this envelope. And there are these glycoproteins are of glycoprotein 120 and glycoprotein 41, isn't it? So now internal to it, there is icosahedral shape which is enclosing the RNAs. Two identical single-stranded RNAs are present, and there is a reverse transcriptase enzyme, which is one of the characteristic feature of retroviridae. And there are some other proteins also, which are essential for its synthesis, protein synthesis, all the things. Okay, like P7, P9. Then moving to the viral genes. That means the HIV viral genes, just we'll have a glance of it, which is also very important. The genome of HIV is going to have three structural genes, okay, that is GAG, POL, and NV. And there are other non-structural and regulatory genes also specific to the HIV. The GAG gene, so here red color you can see, the GAG gene codes for core and the capsid of the virus. That means here is going to be the core and the capsid of the virus is going to be coded by GAG gene. And then coming to the POL gene, codes for the enzyme reverse transcriptase for this one. This is going to be POL. Then coming to the NV genes, decides the envelope glycoproteins GP120 and GP41. So this is going to be coded by the NV gene. So that's how these three are going to be of mainly involved in the structural genes. There are certain other non-structural genes also, like trans-activating genes. Those are called as TAG genes. So here you can see. And then negative factor gene. So that is going to be of NEF. So here the pink color is going to be indicated as negative factor gene, which regulates the viral replication. And then coming to the regulator of virus gene, that is REV. So here you can see blue color one, enhances the expression of non-structural proteins. And then the fourth one is going to be a viral infectivity factor gene, that is VIP gene, VIF, influences the infection property of viruses. So in addition to all these things, it is also going to have some sort of VPU, then VPR, then LTR. Okay, like this, there are also genes are also present. And the multi-ability rate is in HIV is very high, rendering its control over difficulty. As it is going to change its antigenic variations more frequently, it's also very difficult to find out the vaccines, all those things for this HIV infection. Okay, so this is all about the viral genes. So the viral genes is going to have the structural genes as GAG, POL, and NV, and it is also possessing the non-structural genes like TAT, NEF, REV, and VIF. Okay, and altogether it is good that this genome is going to be of having of nearly 9,749 nucleotides. Okay, so that's all about the virus structure as well as the viral genes. Then moving to the multiplication or replication of virus. That means once it enters into the host system, what is happening in our body. The HIV replicates within the infected cell, that is our T4 cell or T lymphocytes, sorry, CD4 cells of T lymphocytes in human blood. After its RNA coming out of the capsid core, RNA molecules of virus acts as mRNA. So this whole thing you can see in the picture, then you'll get the clarity of it. So here is the total replication of HIV infection. See what's happening. So first of all, this is the HIV virus, which is entered into the host cell, and from the blood cell, it will find out its T4 cell. Okay, that is CD4 cell. So this is the CD4 cell. Now the fusion is going to occur with the spikes, that is glycoprotein 120, and here is our CD4 cells. So there will be anchoring between these two. Now what happens? Slowly the envelope is going to degenerate, and now it is going to the HIV RNA and the reverse transcriptase, integrase, and other viral proteins enter the host cell. So this envelope is going to degenerate, and this nucleocapsid is going to enter, which possess all these things, integrase, and then viral proteins, that is P7, all those things, and then single-stranded RNA with reverse transcriptase enzyme. Now once it enters into the cell, host cell, this pre-integration complex is going to be dissolved and the viral genome was released. Now once it is going to release, the viral RNA with the help of reverse transcriptase is going to synthesize the viral DNA. And now the DNA with the enzyme integrase, with the help of this integrase enzyme, will integrate in between the that means in our DNA, that is host DNA. So once the host DNA is going to have the integrated DNA, this stage of virus is going to be called as provirus. What we call it as provirus. So here I have written the viral RNA is first transcribed into a single-stranded DNA and then into a double helix DNA is going to be called as provirus. And the HIV DNA like this is going to be stay for a long time. That means it won't express any type of symptoms that there is an infection in our body. Now after some time, what happens? Slowly this DNA starts up synthesizing the viral RNA used as a genomic RNA and to make viral proteins. So coming outside, it is going to synthesize all the machinery that it requires, and obviously new viral RNA and proteins move to the cell surface and a new immature HIV forms. And now the virus is released. Viral proteases cleaves new polypeptides to create a matured infectious virus. So this is the reason why in the structure we have discussed that the envelope was derived from the lipid bilayer of the host cell. So now the mature virion is ready. Again, it is ready to attack the new cell. And at last, this cell is going to, the infected T lymphocytes producing HIV no longer divide and eventually die. So that's what happening in this HIV multiplication or infection stage, that is replication of the stage. So what is happening? The HIV it replicates within the infected cells, that is like T lymphocytes in human blood. After its RNA coming out of the capsid core, RNA molecule obviously acts as an mRNA. So here RNA molecules acts as an mRNA using the enzyme reverse transcriptase, which is intact virion. Okay. Now the RNA acting as a template to form the complementary single-stranded DNA, which is converted into the double-stranded. So first of all, it is going to be converted into the single-stranded, and then it is going to convert into the double-stranded DNA. Is now transported to the infected cell nucleus, which integrates at specific site into the host chromosomal DNA and exists as provirus. Okay. Now the HIV DNA can remain in this latent phase for long periods. Eventually, however, productive virus synthesis starts and new HIV particles are produced from the infected cells, and the progeny virions are formed and released from the infected cells by the process called as, this process is called as budding. So budding process is going to occur to release the new virion particles. Now what happens? This infected T lymphocyte producing HIV no longer can divide and obviously it dies. So this is all about the HIV replication or the multiplication. Okay, so this is all about the HIV structure and multiplication. Then just have a glance of just how it is going to be transported, and then what is its symptoms, everything you can go through in the medical microbiology. We had the AIDS topic. There we have discussed about all these things in detail. Okay, so in this video we are majorly concentrating on the HIV structure and the replication or multiplication of HIV. Okay, thank you.